Hello everyone and welcome to this session. Today I will be presenting on graph signal processing and a special take on how high resolution connectomics relates to graph signal processing. To give you an overview of today's talk, I will first start with an introduction to graph signal processing and describe some of the main topics of GSP such as graph Fourier transforms and graph signal filtering. I will then move to describe how it relates to current network neuroscience research, how brain network organization, structure, function, and behavior could be related using graph signal processing, how we could represent the bold signal activation as a graph signal, and finally, how to detect structure function coupling using a graph signal processing framework. I will then move on to the topic of high resolution connectomic applications of GSP. I will give a brief description of what a high resolution connectome is and how it is different from normal brain connectivity maps. And I'll then describe what we could do with GSP when we apply it on high resolution connectivity matrices and how we could extract behavioral biomarkers of cognition from high resolution connectomes using GSP. Hopefully, I will conclude today's session with a few take-home messages that you could remember from my talk. Okay, so without further ado, let's start talking about graph signal processing. So graph signal processing is a generalization of discrete signal processing techniques that is being applied to the realm of networks. We know that traditionally discrete time series can be transformed using a Fourier transform to a frequency domain. And this information could then be decoded at a later stage back to a time series. And we could extract back the original signal using its frequency domain representation. Now, similarly, we could have a graph signal, which is basically a signal that is defined over a graph structure. So a set of arbitrary values being assigned to every node of a graph. We could then apply graph Fourier transforms to encode the graph signal into a bunch of frequency domain harmonics. So in the picture here, the very first harmonic on the left is the lowest frequency observed on a graph, and we can see that there are not that many perturbations or alterations in the signal in this lowest spectrum or lowest frequency. But as we go to a higher frequency, we can see that there are more changes in local changes in the values of the signal over the defined graph. So basically, graph Fourier transform encodes a graph signal into the basis of these frequency domains so that a signal will be represented as a linear combination of the spectrum. So graph Fourier transform can be defined using a graph shift operator, and for the purpose of this talk, we'll be mainly focused on the symmetric normalized Laplacian, which is a normalized version of the Laplacian graph shift operator. Now, once we have a graph shift operator, we could use eigen decomposition to provide a basis for graph Fourier transforms. And then any arbitrary signal x could be encoded into the graph frequency domain using a matrix multiplication. And whenever we need to convert the signal back to the graph signal domain from the frequency domain, a similar matrix multiplication can be used to retrieve the original graph signal. Now, after that, we could then apply graph signal filtering to the graph signal that we have collected. And this equation here basically summarizes what a graph signal filtering does. Now, in order to go to the detail a little bit, if we have a graph signal X, we could first encode it into a graph spectrum using the graph Fourier transform. We could then filter this spectrum to select only specific frequencies of it, or use a weighted measure to indicate how much of different frequencies we would like to keep. And once we have filtered this spectrum, we would apply the inverse graph Fourier transform to build the filtered signal again. Now, with that short introduction to graph signal processing, I would like to move on to the topic of network neuroscience and how GSP relates to network neuroscience. So with network neuroscience and connectomics, the brain is basically modeled as a network of edges and nodes. The edges of these networks could be defined either using structural connectivity information, which explain the anatomical connectivity between different brain regions, or the functional connectivity information, which describes the functional co-activations across different regions. Now, in the neuroscience literature, it has been observed that the brain structure and function are closely related. So if we took the structural connectivity of an individual and their functional connectivities, there's a good chance that they will be highly correlated. And graph signal processing is a really good tool to study this structure function coupling. Basically, we use a graph topology that is extracted from structural connectivity to define our graph, and then we use information from the bold fMRI time series to construct our graph signal at any given time. Then we could perform graph signal filtering over this graph signal to decompose the fMRI signal into a low pass and a high pass regime. So we would have a low frequency mode in which the function is mostly coupled with the underlying anatomical structural topology. And we would also have a high frequency mode in which the function is relatively independent of the underlying structure. Now, previous studies have looked into this structure function coupling and have observed that the patterns of decoupling observed across the cortical surface is a lot similar to the principal functional connectivity gradients studied, which shows a higher coupling in regions of higher order association cortices and a lower coupling in visual and motor systems. Now, I would like to discuss about the potential applications of graph signal processing on high resolution connectomes. So to give you an introduction of what these high resolution connectomes are, I should say that high resolution connectomes mainly differ to the traditional connectomes based on their definition of the nodes of the brain network. So traditionally, when we want to construct a brain network, we use a brain atlas, and the delineation of different regions of the brain atlas is what we use to then construct the nodes of our network. However, we have recently proposed an alternative view in which we use the high resolution data available as a delineation for the nodes of the network. So basically, the nodes of the network are the cortical surface vertices or the voxels of the brain image acquired. And we would basically like to apply graph signal processing onto this high resolution connectomic structure. So we mapped both structural and functional connectomes at the resolution of about 100,000 nodes, which constitute both the cortical brain regions using cortical surface, as well as the cerebellum and the subcortex in a volumetric representation. We mapped these connectomes for every individual in the Human Connectome project, and then we intended to perform graph signal processing. So we first computed a group average structural connectome in high resolution, and then we used eigen decomposition to basically compute the first 1,000 eigen components of the graph signal, the graph shift operator. And basically, these gradients that are being visualized right now are the first few eigenvectors of this eigen decomposition. So what we observe is that these eigenvectors can be divided into sets of cerebellar eigenvectors and cortico-subcortical eigenvectors. So I would like to now focus on these cortico-subcortical eigenvectors and basically share one of the interesting observations that we have, and that is that we see that these harmonics or these eigenvectors of the structural connectomes are actually independent of the resolution that we use. So here in this high resolution setting, we can see that the first four main eigenvectors basically constitutes the first eigenvector, which is just the degree distribution of the actual group average structural connectome. The second eigenvector is basically a separation of the left and the right cortex. The third eigenvector is an anterior-posterior separation of the cortex, which is symmetric on the left and right side. And finally, the fourth eigenvector is an asymmetric gradient from the visual regions to the rest of the brain. And what we observe is that similar harmonics or similar eigenvectors were also detected in previous research. For example, this is a seminal work by Atasoy and colleagues, and we see that these left, right, and anterior, posterior harmonics are also present in that work. Even if we go to a lower resolution graph signal processing study, we can see that in this work by Preti and colleagues, we still see the same eigenvectors. So we have the degree eigenvectors, we have the left-right separation eigenvector, we then have the anterior-posterior, followed by the asymmetric left-right, anterior-posterior components. So it is really interesting that these harmonics are actually existing across all these different studies using different resolutions, different structures. This one, for example, is a work by Tarun and colleagues in which the structural connectivity was actually represented in a voxel-based high-resolution setting. And we can see that similar eigenmodes are even visible in this work. And this kind of tells us that these eigenmodes are one of the inherent characteristics of structural connectomes, we could say. Now that we have mapped the eigen decomposition of high-resolution structural connectomes, we could use this eigen decomposition to perform graph signal filtering. So we basically used the high-resolution bold fMRI data, and we found, first of all, the number of eigenvectors that we needed to have to store on average 50% of the signal energy in a low-pass filter. We then created a low-pass and a high-pass filter, and we basically decomposed the fMRI signal into a part of the signal that was aligned to the underlying structural connectivity and a part of the signal that was liberal to the underlying structural connectivity. Then, similar to previous work, we computed decoupling index, but in high resolution. And this plot here basically shows the decoupling index. So regions in red basically highlight regions that are basically increasingly coupled. These are regions, the red regions are regions where we see increased coupling between structural and functional connectivity. So the bold signal follows closely the underlying structural connectivity topology. And the regions that are in blue are regions that are further decoupled so that the functional MRI signal is not actually following the underlying structural connectivity. And similar to previous work, we also see a great resemblance between these decoupling indices and DFC principal gradients showing regions of higher order association cortices. We also see a great deal of resemblance between this decoupling index and the structural connectivity uniqueness gradients, which basically indicate regions of the structural connectome that are unique in every individual. So given that these regions are known to be unique in individuals, we then were wondering if we could use this decoupling information in high resolution to extract behaviorally relevant data and basically predict behavior using this decoupling index. So what we did was we took a sample of only 40 individuals from the HCP data. We looked at their individual decoupling and correlated the voxel-wise decoupling indices with a measure of fluid intelligence, and we used a simple linear model to try to see the associations between high-resolution decoupling and fluid intelligence. And we observed significant and strong effects existing in this small data, but obviously, given that we had so many regions, and given that this is a high resolution data with a large dimension, it is possible that this is due to overfitting. So we wanted to test and see how generalizable this association that we observed was to the greater population. So what we did is we then used the 960 remaining subjects from the Human Connectome Project data set, and we tried to see if the association that we observed in the smaller sample could actually be replicated to the population. And what we observe is that there's still a significant association between the decoupling patterns of an individual's high resolution functional bold activity defined over the structural connectome and their fluid intelligence. And this actually shows a really interesting observation about how graph signal processing applied onto high resolution connectomes can unravel interesting observations about the organization of the brain and how it relates to behavioral variations. So finally, I would like to finish my talk with a few take-home messages, if you will. So basically, in what I presented, I wanted to show that graph signal processing provides a computationally efficient method to analyze high-resolution connectomes. We observed that some of these observed gradients are actually resolution independent. So graph signal processing can be used as a resolution independent measure assessing brain connectivity. And finally, we show that if we apply graph signal processing onto the high resolution connectome, we are able to extract generalizable and reliable biomarkers of human behavior. Thank you very much for listening to today's talk. And if you have any questions, please feel free to reach out to me on my email. Thanks so much.